So hello everyone, this is Chiara, and this is the first webinar of our new series, Nature Nanotalks. In this series, we will try to have a few webinars similar to the one that we're having now, where we discuss topics that might be of interest for our broad nanotechnology community. And today we take advantage of our recent special issue that we published in January that is entitled Nanomaterials for Immunomodulation to kick off this series with 4 amazing panelists who are also the authors of the pieces that we published in the special issue. I will introduce them shortly, but first of all, please let me thank all of you for registering to this webinar. We've had over 350 registrations so far. And the people that register comes from all over the world, so this is great. And thank you also very much for the questions that you put in the registration forms. We will try to answer them hopefully during the discussion, but we will also have some time at the end of the webinar for audience question. And about this, we also have you also have the possibility of sending your questions in through the panel control that you'll see. Hopefully you'll see in your screen, and I'm sharing a slide now. Hopefully you'll be able to see this. So you will have a space for questions. We will collect them and again try to answer them during the discussion or at the end of the webinar. The webinar is recorded, and you'll be able to access the recordings from our website shortly after the webinar has finished. Currently, you are muted, so the only way to ask questions is actually to use the question chat box. Another point that I have to make, we are all working from home, so we are all connecting through the internet, through our internet access, and so this means that there might be the chance that the signal will fail at a certain point. Hopefully this is not happening, this will not happen. If that does happen, we will try to reconnect to the webinar as soon as possible after that, so please bear with us and just click on the link of the webinar again to reconnect with us. So I think that that's it from the logistics perspective. And so now I'm really happy to introduce our 4 panelists. They come from Australia and the US. And so I know that in both cases, the timings is not extremely friendly. So thank you all very much for joining us at this time. Our first panelist is Angus Johnston. He is based in Melbourne. It's very late for you, Angus, so thank you again for accepting to participate. He is based at Monash University, where he is the head of the group working on nanomaterials for biology. And please correct me if I do say something wrong. And his research mainly focuses on developing strategies for targeted drug delivery, and he also focuses on studying the interaction between cells and proteins and nucleic acids and nanoparticles. He is the co-author of a review that discussed the role of nanotechnology for inducing a tolerogenic phenotype on dendritic cells, and this is important. The importance of inducing a tolerogenic phenotype is because it allows to dampen the immune response whenever this is excessive. For example, in the case of autoimmune diseases, but also when we talk about organ transplants. And so having ways to reduce the immune response would help lowering the chances of organs rejection. So this is extremely interesting and important. So thank you very much, Angus, for joining us. Our second panelist is Betty Kim. She is based in Houston, and she is associate professor in neurosurgery at MD Anderson Cancer Center. Her research mainly focuses on developing strategies for cancer immunotherapy that are specifically based on using nanomaterials that target the innate immune system. And we will discuss a little bit more about the innate and the adaptive immune system later on in this webinar. And Betty is the co-author of a perspective that discusses which strategies and which approaches should be used to fill the gap between preclinical studies and clinical outcomes of these studies, especially when we talk about application of nanotechnology for cancer immunotherapy. So really looking more into how to translate preclinical studies into clinical reality. So again, thank you very much, Betty, for being with us today. Our third panelist is Mike Mitchell. He is Associate Professor for Innovation in the Biomedical Department of University of Pennsylvania. His research mainly focuses on developing biomaterials for cancer immunotherapy, but also has an interest in developing vaccines and working with RNA therapeutics. He is the co-author of a review that specifically analyzed which nano-based approaches can be used to improve T-cell based cancer immunotherapy. So thank you, Mike, for joining us. And finally, Ankur Singh, he is Associate Professor at the Department of Biomedical Engineering at Georgia Tech and Emory University. His lab has developed immunoorganoids and strategies that allows better tracking of the fate of immune cells, as well as strategies to help translation of therapeutics. He's also interested in developing materials for vaccine development, both in cancer and for infectious diseases, and also has an interest in developing approaches to overcome microbiome-associated diseases. He is the author of a review on how to use nanotechnology to induce B-cell immunity, specifically in the case of infectious diseases. So again, thank you all for being with us today. It's really exciting, and I'm sure it's going to be a super interesting discussion. One thing I forgot to say before is that we have decided to have a few slides to better introduce specific topics and to kind of guide the discussion, considering that the topic is extremely broad. We thought that having slides might help structuring the discussion. So to kick off today's webinar, I have one big question, which is why should we use nano to induce immunomodulation? Why should we tamper with the immune system, which is already a fairly complex and layered entity, with man-made materials, which themselves add additional layer of complexity to the system? And also, what is specific and what is peculiar about using nanoscale materials? How can they help, and what are the challenges? So with this question, I think I'll leave the floor to our panelists, and I think that Mike maybe wants to start this. Okay, yeah, the floor is yours.
Yeah, thank you, Chiara. So we have an audience coming from all over. So why nano? Why do we need nano in the world of immunomodulation? The most prominent example, to be honest, that comes to my mind right now is something that's on everyone's mind, which is the COVID mRNA vaccines. So it's a little different in Europe. We have AstraZeneca available now for the vaccines, but for those of us in the US, the two COVID vaccines that are approved by Pfizer, BioNTech, and Moderna are both based on mRNA technology that can get its way into cells. But what's the challenge there? The challenge is mRNA is extremely difficult to get inside of cells. It's large, it's hydrophilic, it's negatively charged, it doesn't readily cross the immune cell membrane. And the reason why these mRNA vaccines that are so important to us now are possible is because of nanotechnology. mRNA needs help to get into these target immune cells, and that help is nanotechnology. And specifically, what these vaccines are comprised of is a lipid nanoparticle technology. So these lipid nanoparticles can encapsulate mRNA, protect it from degradation in the body, but also mediate its intracellular delivery into these immune cells that we want it to go to. This nanotechnology enables endocytosis in immune cells, endosomal escape, and ultimately release of the mRNA into these cells to translate that mRNA into, in the case of COVID, the spike protein. So this is an example where nanotechnology is essential, and we would not have these vaccines right now without decades of nanoparticle and nanotechnology-based research. So why nano? This is one example that's really important to us now, but there are several reasons why nanotech and nanoparticles are so important for modulating the immune system. So we can think of this beyond the COVID vaccine. Immunomodulation plays a key role in infectious disease, cancer, and other diseases. And one of the major benefits of nanotech is the size that we could exploit. I remember we had a great conversation with Ankur Singh about the unique properties of the nanoscale. This allows us to get particles that contain therapeutics or vaccines. It allows them to gain access to draining lymph nodes. So the draining lymph nodes are rich with various types of immune cells that we can therapeutically target. And because of the size of these particles, we can efficiently get them to these sites. Their surface functionality is also another major benefit as well. So the surface of these nanoparticles can be exploited to be functionalized with various types of small molecules, peptides, and antibodies. And what that allows us to do is, especially, if we have immune cells that have specific markers on them, homing targets that we could look at, we could exploit this mechanism to allow the particles to latch on to different immune cells and target them, whether it be in the blood, in tissues, or the lymph nodes. Their physical chemical properties are very important. So the types of therapeutic cargoes that are available for us for immunomodulation can vary in their size, their structure, their hydrophobicity, their hydrophilicity. So by being able to tune the physical chemical properties of nanoparticles, we can encapsulate different types of molecules that we might need for immunomodulation. And the last 2 points I'll mention here, which you mentioned, Chiara, are control and delivery, which I kind of think of in this case as somewhat related. So with nanoparticles, what we can do is we could really control the delivery and the release of various therapeutic molecules in the body. Whether we want, say, burst release, maybe in the case of a lipid nanoparticle, when it gets out of the endosome, we want the mRNA released immediately. Or in certain cases where we require prolonged release of a therapeutic over a long period of time for immunomodulation, we could also control the properties of the nanoparticle to do that. And then the last thing I'll mention with control and delivery is nanotechnology really allows us to also control systemic exposure of immunotherapies and immunomodulatory molecules that could potentially be toxic to the body. One of the early examples I think of that was FDA approved in the 90s is a recombinant form of interleukin-2. The challenges that we faced in using that for immunotherapy is significant toxicity and vascular release syndrome that we see in patients due to the systemic exposure of this recombinant molecule. What we've seen in the field of nanotechnology is we could control the presentation of this interleukin-2 in terms of the immune cells that it's exposed to in the body. So we could prevent some of these systemic exposure and toxic effects while still being able to maintain the interactions of these immunomodulatory molecules with immune cells in the body. So these are some of the benefits, but what are some of the challenges? So we make these advancements as engineers in the field of nanotechnology, and we have a lot of tools at our disposal now, but to truly realize the potential, these go hand in hand, both nano and immuno. So with the advances in nanotechnology, we really need to couple this with a better understanding of the various components of the immune system, which I think Betty Kim will be getting into next.
Okay. Yeah, I'll move to the next slide so that Betty can actually expand on this. And I also have actually questions about this that probably will be actually answered in the next slide. My big question, for example, is like, because of the large amount of choices that you actually have, how do you go about picking one, and is one better than another, and is targeting specific cells or specific mechanism beneficial in which cases? So how do you go about with this? However, I've been told that that the slides are cropped, so I'll try to solve the issue. Hopefully I will be able to. Just give me 1 second. Let's see again. Is this any better?
No, it's still cropped at the bottom.
Is it still cropped at the bottom? Okay. Not sure what's going on then.
Sure.
Can you
Chiara,
see?
just
Yeah.
what I can do is just talk about Immunology 101 while you sort out the technical
Sure,
difficulties.
sure. Go ahead.
And then,
Probably it's better.
yeah, that way, yes. So you go on, and I'll just go with the Immunology 101 sort of concept. So for those of you who are not so familiar with the basics of immunology, our body system consists of two sort of branches: the innate immune system and the adaptive immune system. And so the innate immune system is like the first line of defense against pathogens, and this includes that natural barriers that exist in our body, like the skin, the mucosal surfaces, and cells that perform functions such as phagocytosis and killing. And these cells include macrophages, dendritic cells, neutrophils, and natural killer cells. So the innate immune system is active and it's always on, if you will, and it's not really antigen specific. On the other hand, the adaptive immune system, as the name suggests, it needs to be primed or educated before it actually recognizes the specific antigens or of their target of interest. So the adaptive immune system can always also produce memory responses against specific antigens once it is educated. So for example, T cells and B cells are key components of the adaptive immunity, and T cells are responsible for cellular immunity, while B cells are responsible for humoral or antibody-based immunity. The functions of the body's immune system is quite diverse. So for example, although innate immune system plays a very important role in clearing things like infections and like cancer cells, for example, they're also very important in maintaining just regular tissue homeostasis and for detoxification of the body or organs, as in the case of macrophages, for example. We also have to realize that the innate and adaptive immune systems are in fact two different branches, but they're really not that separate. They have they're highly connected. So for example, you need antigen presenting cells that are part of the innate immune system, such as dendritic cells, macrophages, microglia, in the case of in the brain, to present antigens to T cells to educate them. And then the T cells then go on to kill the cells that are infected or diseased, or or it can activate B cells. Similarly, in the reverse, T cells themselves can actually produce cytokines that then can help activate the innate immune cells, such as dendritic cells. So these feedback mechanisms are critical in terms of understanding how they interplay and where they interplay, so that we can actually create better sort of novel nanomedicines when we're designing them for our specific need. So for example, in terms of understanding their interactions and how these certain pathological conditions disrupt these processes, I think will help us become better sort of nanotechnologists, if you will, to design them. So for example, in cancer research, which I am interested in, to generate a system that's an anti-tumor immune system, you initially want to present tumor-associated antigens to naive T cells. And then these T cells will then mature, and they become or transform into these effector T cells. And when the effector T cells sort of encounter the tumor cells down the road that express the same antigen, it then attacks it and kills these tumors. So all of these steps that you see on this slide here on the right, each step alone can be disrupted by cancer cells. And therefore, I think that these are potential opportunities when you're designing nanomaterials as you're trying to restore or even enhance this process. So just in a nutshell here, as you can see on the right, just on the right panel, if there's a cancer cell, an antigen presenting cell will interact with it. It will engulf it, digest it, and pieces of this cancer cell, tumor antigens, will then be presented on the surface of the antigen-presenting cell. And what happens then is that that's when the T cells, which are part of the adaptive immune system, will come in, recognize it, will then be primed or educated, knowing that this is a specific antigen that this T cell will recognize and will then go on to kill tumor cells in the case of T cells, or it will activate B cells to create antibodies, and these antibodies will go ahead and target the infected cells. So others actually have shown sort of different ways of targeting and enhancing this sort of pathway. One is to sort of use a method to engage antigen presenting cells so that they are better at presenting antigen to the T cells. Others are working on ways to enhance T cell function to allow T cells to attack the intended cells or targets of interest. Others are using nanoparticles, for example, as sponges, where I think in certain treatments such as radiation, it causes cancer cell death, and the nanoparticles then, or nanomaterials, will soak up these sort of cellular debris of these cancer cells, and they'll deliver them to these antigen presenting cells. So there are a lot of ways, it's a simple diagram, but each are involved in different sort of steps that we can enhance our nanomaterial design as we're sort of thinking of different ways of designing them. So that's the nanomaterial design in a nutshell, and I'll just stop there.
Thanks, Betty. That was really great. I think that talking about targeting, for example, specific cells, and you mentioned about the possibility of enhancing, for example, the T cell function. So Michael has actually written a review on this and on how nanotechnology can help modifying T cell for T cell based therapy. And so I wonder whether you can expand a little bit on this. And we also had a question from the audience in one of the registration form about how immunotherapy can help getting T cells into solid tumors. And I think this is something that you discussed a little bit in the review. So maybe you have, and of course, everyone that wants to chip in, we're more than welcome. But so if you can kind of like expand more about what you could do to improve therapy for solid tumors using, for example, T cells, that would be a good discussion point, I think.
Sure. Was that a question for Michael or for?
Actually, for anyone who wants to talk about that.
Okay. Well, I think the role of the ability to enhance T cells can come in either as a direct way of enhancing T cell function, or upstream, as I mentioned, through either the antigen presenting component, or allowing cancer cells to engage with antigen presenting cells, and allowing the processes of phagocytosis, which is an area that we're interested in developing. In terms of nanomaterials sort of delivery and design, and how we can get different materials to sort of activate these types. I think that's it's currently for in vivo very technically challenging, but there are different ways of doing that. And I think one of the sort of areas that we're actively looking at, and I think probably many are, is how does virus do this? Virus is in the size of a sort of nanomaterial nano sort of regime, and we're almost using what's already in nature, and using sort of synthetic products to sort of recapitulate what happens in nature, if you will. And I think it's sort of a broad sort of answer to a very complex sort of question, if you will, because there are different ways of targeting. You can target it all from the innate, which is why we're focused on innate, because we think it's upstream of what happens downstream. Or a lot of the cancer immunotherapy right now is targeted on T cells, and how we can sort of allow immune checkpoint blockade to work better specifically on T cells. So I think this is like a complex sort of question that I think there are a lot of ways to address. I don't know if the other panelists have additional comments to make.
Yeah, I guess I'll just add to Betty brought up incredible points, Chiara. I think there's a lot of exciting research at the interface of nanotech with T cells, but specifically to overcome biological barriers to solid tumor immunotherapy. I'll bring up two very recent examples of the audience. One of them combines what I mentioned earlier, an mRNA vaccine approach with an engineered T-cell immunotherapy. So very recently, about a year ago, a study was published where we could infuse what are known as CAR T cells, which we haven't gone over too much, so chimeric antigen receptor T cells, in combination with a messenger RNA vaccine. And the reason to do this is to boost T cell expansion, to generate an solid anti-tumor response within the body. So there was a beautiful example recently of where we could deliver an mRNA vaccine that could target these antigen-presenting cells. And when you infuse the CAR T cells into the body, these dendritic cells can then activate these T cells and basically increase the expansion of CAR T cells in the body to generate a solid tumor response. Beyond that, we've seen a few other approaches for targeting solid tumors as well. I believe Betty mentioned this as well, but solid tumors can have a dense extracellular matrix. So there could be several challenges with just getting T cells to traffic across these barriers in the body. We've seen numerous examples in the literature where you can actually develop nanomaterials that could target these fibrotic barriers and actually degrade extracellular matrix, and this could improve the infiltration of T cells within solid tumors. We've also seen the generation of what's called, and these are in clinical trials now, these are called bites. So bites are basically particles, or nanobites are basically particles that could have antibodies that target both the cytotoxic T cell in the body and the cancer cell. So this could be a way of promoting interactions between solid tumor cells and cytotoxic T cells within the body. And then the last thing I'll mention for this in solid tumor immunotherapy is leveraging the microenvironment to improve T cell infiltration. So we're seeing emerging examples in the literature now where we could use that tumor and exploit it to produce different types of proteins using nanotechnology that could turn what we call cold tumor into a hot tumor to promote T cell infiltration.
That's fantastic. Thanks so much, Mike, for this overview.
I wanted to just give an example, specific example of how, in terms of getting T cells recruited and work better. One way, I think it's like twofold. One is to have the T cells transport into the tumor itself. And then one is to make sure that T cells not only survive, but they proliferate and they enter the tumor site of sort of interest. So one of the things that we're working on, for example, is that, what signal pathways activate T cells, and that include innate activation. So for example, CGAS sting is a really cool pathway where you can allow T cells to sort of be activated and proliferated sort of in a way that's more effective within the tumor microenvironment.
Thanks, Betty. So as you as we are discussing, I have like some other questions coming to my mind. I realized that we talk a lot and probably because of us very good reasons about cancer immunotherapy and about how nanomaterials can be applied for to tackle cancer therapy. However, there are other aspects of modulating the immune system, and there are other applications of modulating the immune system that can be considered. And as I was discussing at the beginning, one such example is actually to do the opposite. So instead of boosting the immune system, actually dampening down a little bit and dampen the answer, the response of the immune system towards in specific conditions. So one such conditions can be, as I was saying, autoimmune diseases, and in these cases as well, you do try to target specific cells. In the case of what Angus has written in his review, these are dendritic cells, which are also used to actually boost the immune system. So how do you balance, and what are the the approach is to reach a good balance. What about the materials that they are used? A lot of time, the same type of materials with minor tweaks are used to achieve very different outcomes. So, can you comment on that, and how would you do that? Angus, you can start.
Yeah, it also connects to the next slide that you have, Chiara. So maybe bring to that. That may help the audience to understand.
Can you see it now?
Yes.
Yeah, fantastic.
Angus, would you like to go forward, or?
Oh no, maybe you can talk about this, and then I can jump in once you've introduced the concept of self and foreign.
Yeah. So the slide covers two topics. One is the targeting of the immune system, and the second is the essence of immune recognition, where Chiara was just speaking about recognizing the self versus foreign. And I think now, over the past decade, looking at what the science has revealed, the nanoparticles have a special place in modulating responses in autoimmune disorders. So, as Chiara was referring to, it's related to the suppression. And what happens in an autoimmunity is that your body is targeting your own antigens, your own proteins that they are supposed to not target the way our body evolves and is programmed right from the beginning. So T cells and other cells shouldn't really attack your own cells. But unfortunately, there are diseases such as lupus or rheumatoid arthritis and several others, multiple sclerosis, where your body starts attacking in them. And so and this is in slight contrast to the cancer, where cancer is also your own cell, but now the proteins are an outcome of certain mutations and all. So really using the power of nanotechnology to do it in a way that you can suppress in case of an autoimmunity versus boost in case of a cancer immunotherapy is the beauty of the system. So, for example, if you want to suppress immune system in case of autoimmunity, one of the approach is to deliver small doses of allergen or the antigen to the body. If you simply take a bolus suspension of antigen, you have no control over how much your body is going to. You can, whatever dose you give, that entire dose at once is administered to the, or is available to the cells, dendritic cells and other cells that are responding to those antigens. But by using a nanoparticle, you can tune the release kinetics of those immunogens, and that can induce a more of a tolerance in your body. Now, very recently, there was an article from Dr. Susan Thomas and colleagues, where it shows about how you can tailor the physiochemical properties of nanoparticles and make them degrade in certain compartments of the tissues where you desire these components to be delivered. So they are meant for cancer immunotherapy in that particular application, but I can see their use in autoimmunity as well, and a variety of other places. There are other approaches. For example, you can take the same particle and deliver low doses of antigen along with an immunosuppressor. Now, the key over here, I'm always a big believer of this, that the same cell that is eating up your particle with an antigen should also be receiving the immunosuppressor or immunostimulant. So if you take two different solutions and as a bolus and you administer, you may not be able to target the same cell, but by using a nanoparticle, you can target the same cell. And in this case, an example of immunosuppressant is such as rapamycin. It's an mTOR inhibitor, and it induces a tolerogenic dendritic cell phenotype when you deliver it to the body. And as an outcome, it induces a specific kind of T cell stimulation called regulatory T cells. It makes those T cells differentiate and induces an antigen-specific immune tolerance that is resistant to the pro-inflammatory cascade that is triggered by triggered in the body in the case of an autoimmunity. Likewise, there are several other approaches. So you can do that by taking antigen and tagging to red blood cells. You can take liposomes and co-present an antigen with a ligand that is suppressing specific receptors such as on B cells, which is the case for rheumatoid arthritis. So there are several of these approaches that are out there now and are really making a huge difference. I always teach a topic in my immunoengineering course where I show how rapamycin could be combined with PLGA particle system and can be targeted to the lymph node, which I'll get to in a minute, and has led to a remarkable improvement in multiple sclerosis. This is the work of Dr. Chris Jewell at University of Maryland. And it's so they're very important advancement that nanoparticles bring to the table as compared to other approaches that might be there. Angus, would you like to comment more on that? And then I can wrap this up by specifically talking about the targeting of the organs.
Yeah, sure. And yeah, sort of agree with what you said. And I think getting back to Chiara's question about what materials do and how a particle can be easily repurposed, it's sort of one of the serendipity things that we quite often see is that you might formulate a nanoparticle that you think was going to be very good at antigen presentation and to give you an immunity response. But then when you start to see the response that you start getting from the system, you actually see that it's more looking like a tolerogenic response, and that it's that sort of serendipitous thing that, well, maybe we can go back and re-engineer this material to actually be more useful for tolerance. It's sort of like the first generation of LNPs. The first drug that came on the market was targeting the liver. It was targeting the liver because that's where the nanoparticles went. And so there's a lot of that in the field, is that if your material does something like that, then you sort of you follow that path. But then I think as we get more sophisticated, and I'll touch on it when I talk a bit about my slide, is that if you can see that the native particle is doing something to a small extent, say it might be promoting tolerance or it might be promoting immunity, the key thing then is for us as material scientists is then boosting that natural response that the particle might be doing and knowing what co-stimulatory factors might be involved in inducing tolerance versus immunity. As Anka said, that you can then start loading all of those into the one particle, and the power that you then have with the nanoparticle is that everything is being delivered to the one cell. So you're not stimulating tolerance somewhere where you haven't got your antigen, and then you have side effects that you really want to try and avoid.
Yeah, and to bring you back to the targeting of these immune system, I think there are many ways you can approach, but one example that I want to highlight is, let's say one of the key organ system to target is a lymph node, but accessing lymph node in a traditional way, you're relying on your particles, your vaccine and other drug delivery system to be eaten up by the peripheral antigen presenting cells and then transporting some of that to the draining lymph node. But you can be more efficient in some ways by directly targeting those organ systems. So for example, by Mike brought this up right in the beginning, that by taking a specific size approach, let's say around under 50 nanometer particle size or even smaller, you can directly exploit the lymphatics that connect to the lymph nodes and target that. There was a beautiful paper several years back where this was demonstrated, and since then has been exploited many times. Now, it's not just the size, the functionality of the particle, what's presented on the surface in terms of the chemical groups can also dictate where the particle is going to get transported. And even if it is through the dendritic cell that is getting transported or directly reaching to the lymphatics, one of the major hurdles that these particles see is once they're in the lymph node, they have to interact with specific cells. And so if they go in and if they want to access, let's say, B cells, which are the antibody-making cells in our lymph node, a large amount of research shows that the particles do not directly enter the subcompartments where these B cells live. They do reach the lymph node, but they are not able to enter inside. So one of the recent advancement is from Dr. Darrell Irvine's lab that was published in Science 2 years back, I believe, that you can modify these nanoparticles of certain size, about 40 nanometers, around that size, with nanos or glycosylate these particles. And that really allows these cells to start accessing the deeper compartments of the lymph node. So several of those, so you can't really get that simply by taking an antigen or just taking any particle. You have to do some sort of manipulation of the particle, decorated with certain functionalities for it to be able to reach those targeted compartments. So I'll stop at that with giving a flavor of that. I'm happy to answer things related to that.
Yeah, yeah. So we are receiving actually questions, but I think I'll probably keep them for later on. So another, so talking about accessing the B cells and the lymph nodes, I think this is particularly important for vaccines, for example, and for enabling the development of, for example, memory in the response to infectious diseases, for example. And talking about vaccine, it's of course it's a major topic for immunomodulating materials, and we talk about vaccines for cancers and vaccines for infectious diseases, and clearly these are two very different stories and very different things. Still, we call them the same way. So I was wondering whether you can comment a little bit more on the different processes that will get you to develop vaccines against cancers or infectious diseases. So what are the differences? Obviously, there are antigens involved and antigen-presenting cells in both cases. But if you can expand more on this topic, I think it would be interesting to clarify.
Yeah, I can probably say something a bit about that, and it'll lead into some of the things I'm going to say as well, in terms of depending on whether, say, you're going for a prophylactic vaccine against something like COVID, which is obviously in the news at the moment, or you're going for therapeutic vaccines against cancer, or even the tolerogenic response you're trying to induce. It's about understanding what the response needs to be and engineering the material for that particular response. And I suppose one thing that I think nearly every vaccine on the market is actually, maybe do you want to jump to my slide, because it talks to that a little bit, that the majority of vaccines on the market will be about generating an antibody response. And so we know that, and Betty gave a really good introduction to how that happens, but we know antigen-presenting cells are, in particular dendritic cells, are a major target for delivering our antigen. But equally important as delivering your antigen to those antigen-presenting cells is what those antigen-presenting cells then go and do with it. So when we're talking about the targeting that Anke was saying, that you can target at the organ level, but you can also, within that organ, there's specific subsets of cells that you want to target, and then going even further. Mike alluded to it, and I'll mention it a bit more, is that even then at the subcellular level, there's the place that you want to target the delivery of your cargo to. And all of those are important depending on which particular application you're talking about. And so in particular for, I suppose, the more, to call them traditional now seems a little bit strange, considering how quickly things have moved in the last year, with traditional being subunit type vaccines as against the mRNA vaccines that we've already mentioned. But the traditional subunit vaccines they work by being internalized by these antigen-presenting cells, and they go into the endolysosomal compartment, and you have sampling by MHC2 in those endosomal compartments. And so part of the reason why we have most of our vaccines being based on antibody response is partly because it's relatively easy for us to deliver material to those endolysosomal compartments, because that's where they naturally go, and that's where a lot of the natural immune response comes from, and that sort of comes from us learning from nature and letting the system do what it wants to do. And so we can generate those antibody responses relatively well and reasonably robustly. The challenge becomes more difficult if we want to develop those cytotoxic T cell responses. And that's where the antigen is being loaded onto MHC1. And MHC1 loading occurs in the cytosol. So that means that the antigen that's delivered to the cell not only needs to get inside the dendritic cell, but it needs to get from the endolysosomal compartment out into the cytosol. And in our discussions with Kara beforehand, I said I wouldn't get too controversial on this area, but I would say our understanding of endosomal escape is relatively poor at the moment. And being able to understand those subcellular processes more, I think, is going to be really key for improving all areas of not just immunomodulation, but for a whole range of things, because relatively few of the biological therapeutics we're trying to deliver are active in the endosome. Most of them are active elsewhere. And I think it's an area that I'm interested in trying to quantify how efficient these processes are. And as was mentioned earlier, can we learn from biological systems? Can we learn from even a virus like COVID? How efficient is it at delivering its genetic material into the cell? And it might be that COVID is 1% efficient, because our best estimates at the moment is our LNPs. They're probably only about 1% efficient at delivering the mRNA that gets into the cell. 1% of it is getting and being actively transcribed. The fact that they work means that they're incredibly potent based on very inefficient processes at the moment. And that, I think, is one of the real sort of areas that nanoparticles can look to, is if we can start understanding these cellular processes and harnessing them. If we're at 1% efficiency and we're developing these vaccines that work, if we can go to 50% efficiency, then you lower your dose significantly, you lower the cost, you lower the side effects that you have associated with some of the excipients that are part of the formulations. And so I think that's definitely somewhere where the outlook is really interesting for material science formulations. Jumping a little bit in terms of delivering, whether it be that antigen, like protein antigen to the cytosol, or if it's an mRNA, it needs to get into the cytosol to be translated into a protein. An alternative approach in terms of vaccines is to target subsets of cells, so subsets of dendritic cells known as the cross-presenting dendritic cells, and they're known to be able to take exogenous antigen, so they'll take antigen from outside the body, and they're outside the cell, and they're able to load that onto MHC class 1 through mechanisms that we're starting to understand, but I still don't think we fully 100% understand. And so an alternative technique, rather than trying to engineer our particle to be able to fight against that endolysosomal pathway, an alternative technique is to target to the cell, which has its own processes that enable us to do that. There's quite a few other things that in terms of nanoparticle engineering for sort of which route we're trying to go through for therapeutic vaccine to tolerance, which relates to the co-stimulatory factors, but probably don't need to go into that now. It might come up in the discussion, but that's probably a good point for me to stop and let others say things.
So if I may just chime in for a minute, I just want to emphasize one more thing here, that for the infectious disease vaccine side, that when we administer vaccines, the antibody responses needs to be and the cells that are producing, they need to be characterized or evaluated in certain ways so that we are sure that what we are getting is very specific, robust, and long-term response. So you can get initial antibody responses, which is, if you put that in the context of COVID-19, yes, you can get an initial response. But for you to get a durable long-term response, you have to go through this particular specialized pathway of B cell maturation that is dependent on the CD4 positive T cells initially. So if your vaccine, nanovaccine, is not leading to enough of the CD4 positive subtypes, these are called follicular T helper cells, but a smaller population than more abundant CD4 in general. But if that process doesn't take place, then you don't have enough required changes or the mutations in the B cells to make antibodies that are very strong, neutralizing, or depending on whether you want a broadly neutralizing versus a more direct monoclonal response. A lot of that is dependent on how your vaccine is received by the body. And I think nanovaccines can control that part. For example, I brought up the glycosylated approach. That definitely shows that you can generate a large number of these specialized B cells as compared to a non-glycosylated nanovaccine, both delivering the same kind of antigen in that case. So focusing on that is very important. And I think moving forward in the materials-based nanovaccine approaches, carefully evaluating those responses and documenting is very important, instead of simply just looking at the endpoint serum titers of those antibodies.
Yeah,
I think.
I want to just talk. Oh, go ahead. Sorry.
No, you go. Sorry.
No, I just wanted to sort of make a comment about, potentially cancer vaccines. I think, we know that lymphatic drainage in cancers are very different from normal sort of tissues. So in terms of like lymph node is like one of the first echelons in tumor spread. So when we're looking at tumor spread or distant metastases where the tumor is now spreading to different sites of the body, they access the deformed lymphatic sort of cancer network first. And so, it will require like individualized nanomaterial design in terms of modifying their surface chemistry, size, et cetera, but also like their material sort of physical properties like stiffness, for example, will be very important. In terms of like developing these vaccines for cancer, I think we really need to sort of generate, it's something that you alluded to, Chiara, is like a very potent T cell responses against our particular target of interest while minimizing like normal tissue toxicity and sort of adverse effects. So one of the key aspects of actually designing a really good cancer vaccine is to find the right antigen. But in the case of tumor vaccines, the antigen has to be present in all cells, which can be difficult because tumors are very heterogeneous at times. And so if we can't target each of these sort of tumor cells, then there'd be a potential for this escape mechanism where certain cells we may not be able to eliminate them, and so they'll recur at a later time. And secondly, a vaccine can only work if the antigen presenting cells can uptake sufficient amount of antigens and present them to T cells. So I think these are all sort of the areas where nanomaterials can sort of improve, and we can sort of modify them to deliver not just one antigen, but a cocktail of antigens. So we sort of broaden the potential immune response. So it's not just specific to one, but multiple. And that's one of the main limitations of CAR T cells and even BiTE that Michael was talking about, and that current immunotherapy just target one at a time. And if there's a mutation or if it doesn't work, we have to now go back to the lab, create another CAR T cell and deliver it. And so I think these cocktails where we can sort of load a lot of these antigens at one point and give it simultaneously is a advantage. So I think, the nanomaterials can augment a lot of those processes, but it's very complex as we're all sort of trying to discussed today, that it's a very complex steps that are involved.
Yeah, and sorry, Mike, go ahead. And then I think there is something related, a question that came from the audience that is related to what Betty was talking to. You might want to expand a little bit more, but go ahead, Mike.
And I'll just add, Betty brings up some great points about cancer vaccines. And of course, I'm biased. I'm going to bring up messenger RNA technology again. But, when we're thinking about, vaccinating against cancer and, finding the right types of antigens, a huge benefit of some of these, recent technologies for messenger RNA delivery is if you do a back of the envelope calculation, you can encapsulate anywhere between 8 to potentially 10 different mRNAs within a single nanoparticle. So going back to what Betty said, if we're targeting some sort of neoantigen or we need to target against multiple antigens, we now have the technology to do this on the nanotech side, but also on the mRNA side as well. We could rapidly develop some of these cancer vaccines simply by altering the sequence of these mRNAs to potentially develop something that's really tailored to a patient's tumor.
And I think on the point of LNPs and COVID, I think what we're going to see is because LNPs have leapt forward, I think faster than anyone you probably expected during this crisis. It means that what we're learning from the Moderna and Pfizer vaccines are going to very quickly translate to other areas that we wouldn't have had human data in for 5-10 years in the normal situation, but now that we're going to have that data for the LNPs, I think they're really going to be one of the major nanoparticles moving forward.
That's great, and that's actually answers a few of the questions that had been sent in. So one of the questions that came recently, we have in theory we have 5 more minutes, but I think if it's okay with you, we can go a little bit over the 1 hour time if you have time to stay with us, just to touch upon a few topics that came out during the discussion and maybe just wrap up with perspective and outlook part. So one of the questions came in is from Kyle Schuler, and so it says, because of the constant mutations that cancer cells go through while they proliferate, so how can nanotechnology help overcome the challenge of having different surface proteins on cancer cells all the time? And another question that came in is if you could share your opinion on how immunodominance might affect when you have multiple antigens. Hello, can you hear me?
Yes, I just don't think anyone was immediately jumping in, but I can jump in terms of the answering for in terms of the being able to fight the mutations, and I think Michael probably touched on it earlier, was that the advantage is that if you're, say for example, you're going down the mRNA path, what you have is a biochemically identical nanoparticle. All you're changing is the sequence of mRNA, and so if you start being able to detect that you have particular mutations and you need to raise antigen against that, then your formulated nanoparticle is identical. The sequence is different, and so you need to make sure you understand that you're expressing another protein, which is obviously going to change the immune response. But the big advantage of nanoparticles is that you can have both the multiple antigens and also potentially be switching in and out antigens as it would be for mutations in cancer, but also mutations in something like COVID. If we see that there's these more resistant mutants starting to come forward, then it's only a change of a couple of bases. It shouldn't change the performance of the vaccine. The difference is the antigen that's being delivered.
Fantastic, great. Thank you so much for answering this. And another question that came in from Roy van der Mel. So he's wondering about the LNP, for example. If you think that next generation RNA vaccines will contain different components that substitute the PEG lipid, considering the adverse immune reaction that has been documented.
So I could take that one. So that's a great question, Roy. We've definitely seen, in the news that we've seen, some reactions to the LNPs, and this could potentially be due to the PEG lipid because we've seen, the generation of in various animal models and humans, the presence of these anti-PEG antibodies. So I think it is something to be aware of, and this is something that we need to monitor. But what I will mention is with any sort of vaccine, we do see allergic responses to various patients to different types of vaccines. This is something that happens. And with the PEG lipid, we've seen these responses in patients, but one thing I'll mention is this is a very small subset of patients. This is something they observed initially, and it was a certain percentage of the population, but actually as we've moved on and we've dosed more people with this vaccine, this percentage gets lower and lower. So it's a very small portion of this population. But, with that said, Roy, I think we do need alternatives. So we need to at least investigate potential alternatives to lipid PEG. There could be several benefits to this. One thing that we could potentially do is reduce the allergic response to these LNPs in a small subset of patients. But broadly, when we're thinking about LNPs for immunomodulation, the presence of PEG antibodies can cause these LNPs to be cleared from the body. And, not so much the allergy response, but these could become less effective and they could become less potent as we redose the LNPs. And we're starting to see some work on this. There's been some recent papers in the literature. Instead of, having these lipid PEGs, they have these lipid peptide conjugates that acts very similar to a lipid PEG. So I think this is a very interesting area of research, and I think over the next 5 years or so, we're going to see more alternatives to this because, if we move more into the messenger RNA space and more people rely on LNPs that consist of lipid PEG, it's definitely worthwhile looking at some of these alternatives that are emerging.
I think it's also important to note as well that it's not just limited to the LNP type formulations that you see this, and in fact it's probably more of an issue for something like the AstraZeneca vaccine, is that you get a response against the adenoviral proteins. And so I think from certainly not my expert understanding, but the reason why the low dose of the AZ vaccine was probably more effective was because that they were lowering the response against the adenovirus rather than actually what was happening with what they're delivering is associated to the adenovirus. So I think PEG, it definitely has that potential issue, but I would say it's probably less than something like adenovirus.
Great, thanks so much. I think we are close to wrapping the discussion up. One point that I wanted to discuss a little bit with you, and we had one question from the audience as well, is about translation. And so how far do you think we are from real translation? And the question from the audience came from Christine Horst, and she's asking how much patient-specific or disease-specific design will be required to allow translation of the materials.
Translation's always the really tricky question, but I can't remember it was in one of the Nature journals. I guess it was Nature Immunology, when they were lining up what the potential sort of first off the rank vaccines was going to be for COVID, and they were doing pros and cons of everything. And LNPs came in as a tick for just about everything except translation. They said the big problem with the LNPs is that they're not going to get approved in time, and they'll probably be the last of all the vaccines that get approved. I think that came out in June or July, and within 6 months, LNPs had won. And so I would say, like I mentioned earlier, there's been almost an order of magnitude step in the advancement of LNPs because of the situation that we're in. It's clearly going to be slower when it comes to translating some of the things that we've been talking about today. When you're not in a pandemic, and particularly with everything in the immune system, making sure that you're not having any off-target effects is critical. And I think certainly for the tolerance work that was in the review that I co-authored, there's still a long way to go before that's going to be, well, I mean, some of it is in translation at the moment before there's widespread adoption of these techniques. I think there is still a way to go. But in terms of if I'd been asked a year ago where we would be in translation of them as many things, it certainly wouldn't be where I would have said now. So sometimes these things can jump forward, and you have advancements that come through other fields that then allows that step change in another field.
Well, connected to translation is also whether the pharmaceutical companies will see manufacturing as an angle that is easily possible with these vaccines. So when you come down to synthetic vaccines or particle-based vaccines, and if you're combining with antigens and adjuvants, then you have to develop a pipeline where you can do the GMP-like production and validation, all of those things for individual components. And unlike a virus-based vaccine, these are not going to self-replicate and increase in the amount. And so it does impose some challenges from that standpoint as well. That goes into the calculation of whether how much of effort is there in pushing these for translations, for translation.
Okay, great, fantastic. Thank you very much for this. Thank you very much for this amazing discussion. I really enjoyed it, and I hope our attendees enjoyed it too, and I hope you enjoyed it too. It was really great to have you all with us, and thank you very much.
Thank you.
Thanks very much
Bye,
for
guys.
organising. Good. Thanks.
Bye.
Thank you. Bye.